If I ask you to picture evolution, you probably picture like a bird's beak changing shape to crack a harder nut.
Right, or a fish growing a sharper jaw.
Exactly. It's external,
Yeah.
you know, it's obvious. You can hold it in your hand or just measure it with a ruler.
Yeah, you can actually see it.
But today, we're looking at this staggering data set from a new paper in Nature that proves the most aggressive, rapid evolution on Earth isn't happening where you can easily see it.
No, it's not.
It's actually happening inside the dark, microscopic, kind of twisting landscape of the gut.
Yeah, I mean, we are stepping completely past the teeth and the jawline here, because the reality of biology is that catching the prey is, well, it's only half the battle.
Right.
The evolutionary pressure doesn't just stop once the food is swallowed. In fact, you could argue that is where the chemical and cellular battle really begins.
And to explore that hidden frontier, we're doing a deep dive into an incredibly dense piece of research today. It focuses on 24 hyperdiverse cichlid fish species from Lake Tanganyikan in East Africa.
Plus one outgroup, the Nile tilapia.
Right, the tilapia. So our mission today, alongside you listening, is to figure out how evolution physically rebuilds the digestive tract. Literally cell by hardwired cell, just to squeeze every ounce of survival out of a shifting environment.
A massive undertaking.
It is. And to start, I want to talk about the sheer scale of how they mapped this, because, I mean, if you just grind up a fish intestine, you just get a biological smoothie, right?
Yeah, essentially.
So how do you map the microscopic architecture across 24 wild species?
Well, you have to abandon that smoothie approach entirely. The researchers use a technique called single-cell transcriptomics.
Okay, unpack that a bit.
So instead of looking at the bulk DNA or RNA of the whole organ, which just gives you an average, they use microfluidics to isolate individual single cells.
Like one at a time.
Literally one at a time. They capture one single cell in a microscopic droplet, they break it open, and they sequence the RNA just from that specific cell.
Wow. And RNA, of course, being the messenger that tells us which genes in that particular cell's DNA are actively turned on or off.
Exactly right. By looking at the RNA, you see what the cell is actually doing, not just what it has the potential to do.
Wait, so they are doing this one cell at a time for an entire digestive tract?
I mean, mechanically, yes. It's fully automated through highly advanced sequencing technology, obviously. But they didn't just do a few. They sequenced 82,621 individual cells.
82,000 cells. That is wild.
Across all these species, yeah. And by looking at the unique combination of genes turned on in each of those cells, they identified 24 entirely distinct cell populations just living inside the cichlid intestine.
That level of resolution is, I mean, it completely shatters the way we visually think about intestines.
Oh, absolutely.
Because if you look at human anatomy, we have a stomach, we have a small intestine, a large intestine. We have very obvious physical compartments.
Distinct zones, yeah.
But a cichlid gut, if you just pull it out and look at it, it's basically just a long, plain tube. Yet this single-cell data proves that internally, structurally, it is not a uniform tube at all.
Right. The physical anatomy is completely deceptive. The cellular map reveals profound subfunctionalization along what we call the anterior-posterior axis.
Meaning the front and the back.
Yes, front to back. The front of the gut, the anterior, is fundamentally different on a molecular level from the back, the posterior.
Okay, so how are they different?
Well, in the anterior section, the tissue is heavily populated with a specific cell type the researchers identified as enterocytes 1.
Enterocytes 1, got it.
These cells are essentially microscopic fat processing stations. They are highly specialized, turning on specific genes for lipid and small molecule metabolism.
I was visualizing this when I read the paper like a factory assembly line hidden inside a really plain brick building.
That's a great way to picture it.
Right, from the street, it's just a long building. You'd think the whole thing does one job. But inside, Station 1, right by the loading dock, is entirely dedicated to processing fats.
And as you move down that assembly line to the back of the building, the posterior, the entire cellular workforce turns over. Back there, the tissue is packed with what are called lysosome-rich enterocytes, or LREs.
LREs, and lysosomes being the little recycling centers or chemical stomachs inside a cell.
Exactly the right way to think about them. Lysosomes are packed with enzymes designed to break down really complex molecules. So these LREs in the back of the gut are heavily specialized for protein absorption and the intracellular digestion of complex proteins.
Okay, so I want to pause here and do a quick 5-minute recap of what we've built so far.
Go for it.
So, bullet point 1. We know this cichlid gut is heavily zoned internally, even if it looks plain on the outside.
Yep.
Bullet point 2. It's fats in the front, with enterocytes 1 and proteins in the back with the LREs. Which brings me to a question for you, the listener. Think about the engineering of it all. If you were designing a biological factory from scratch, why might it be vastly more efficient to process the easily absorbed lipids and small molecules right away at the front door and then save the heavy-duty, enzyme-intensive protein breakdown for the end of the line?
It's a great engineering puzzle, and having that invisible baseline map, knowing exactly where these cells are supposed to live, is what allows us to ask the next much bigger question.
Because this baseline isn't static.
Exactly. The researchers wanted to know how millions of years of changing diets completely redrew that cellular map.
Which brings up a massive logistical problem. They're looking at 24 different wild fish species that eat wildly different things, right?
Right, yeah. Algae, insects, other fish.
Some even survive exclusively by biting the scales off of larger fish, which is horrifying but cool.
Nature is brutal.
It really is. But it's not like these wild cichlids have dietary food logs. How do we know what their ancestors have been eating for the last few million years?
Well, the fish leave a permanent chemical signature in their own tissues through what are called stable isotopes.
Okay.
The researchers specifically looked at the ratios of nitrogen-15 and carbon-13 in the muscles of these fish.
Let's unpack the mechanics of that, because stable isotopes are basically nature's receipts. But how does nitrogen-15 actually tell you if a fish is a vegetarian or a carnivore?
It comes down to bioaccumulation. Nitrogen-15 is a slightly heavier, stable version of regular nitrogen.
Right.
And because it's heavier, it doesn't get excreted from the body as easily as lighter nitrogen does. It sticks around.
Oh, I see.
So an algae eater eats a plant and takes on a baseline level of nitrogen-15. But when a carnivorous fish eats that algae eater, it inherits all that heavy nitrogen plus whatever it accumulates itself.
So it concentrates heavily as it moves up the ladder.
It concentrates very heavily. So the ratio of nitrogen-15, which they denote mathematically as delta 15N, acts as a direct proxy for the fish's trophic level.
Meaning where they sit on the food chain?
Exactly. A low number means you're looking at an herbivore. A very high number means you're looking at an apex carnivore.
Okay, and what about the carbon-13?
Meanwhile, they use the carbon-13 signature as a proxy for the fish's habitat. It distinguishes if they feed down in the muck near the shore, the benthic zone, or if they hunt out in the open water, the pelagic zone.
Got it. So they compile these stable isotope receipts to figure out exactly where the fish sits on the food chain, and then they cross-reference that with the factory assembler line, the single-cell map we just talked about.
And the correlation they found is massive.
Right, because the abundance of those fat-processing cells in the front of the gut, the enterocytes 1, just skyrockets in the carnivorous fish.
Yeah, the fish eating a high-protein, high-fat meat diet have literally rebuilt their internal assembly line. They feature far more of these specific fat processing stations compared to the algae eaters.
The carnivores essentially expanded the first phase of their factory to handle the massive influx of complex animal lipids.
That's exactly what happened.
Okay, but I have to push back here, just to play devil's advocate.
Sure.
How do we know this is actually hardwired evolution? Let's say I suddenly adopt a strict high-fat keto diet for 6 months.
Right, your body reacts.
Exactly. My body's going to react. My gut might temporarily upregulate certain enzymes or maybe even adjust the cell ratios to handle all that extra fat. But if I switch back to eating only apples, it resets.
It's phenotypic plasticity.
Right. So how do the researchers know this isn't just short-term plasticity? The fish's gut is just reacting to whatever it happened to eat that month in the lake.
It's a crucial question, and the researchers anticipated that exact critique, which is why they didn't just rely on the RNA expression levels of wild-caught fish, which, like you said, could fluctuate.
So what do?
They validated the actual physical architecture of the tissue. They brought in a much higher level of morphological evidence using a technique called fluorescent in situ hybridization, or FISH.
Essentially making the specific genes glow in the dark.
Making the RNA transcripts glow, yes. They took a specific marker gene called AMCase1, which basically acts like a homing beacon exclusively for enterocytes 1.
Oh.
They designed a complementary probe attached to a fluorescent molecule. When they wash this over the gut tissue, the probe chemically binds only to the AMCase1 RNA.
And then you put it under a microscope.
And under a microscope, every single enterocyte 1 physically lights up. They did the same thing with a gene called CUM to light up the protein-digesting LREs in the back.
Okay, but how does a glowing cell prove it's a permanent evolutionary change? Couldn't there just be more glowing cells because the fish ate a heavy meal yesterday?
That's where the comparative anatomy and lab controls come in. They controlled for diet in a laboratory setting, they raised them on uniform diets, and the structural differences persisted anyway.
Ah, I see.
It wasn't just that the cells were, quote-unquote, turned on more. The actual physical proportion of the gut tissue dedicated to these cells was permanently altered. The architecture is locked into their genetics.
So it's not a reaction, it's a blueprint.
Exactly. The carnivorous species have evolved to maintain a much higher permanent baseline proportion of enterocytes 1 to efficiently process high-fat diets. And they often do this over much shorter overall gut lengths than the herbivores.
Okay, so let's do another quick recap.
Let's do it.
Bullet 1. We track their diets using isotope receipts, the N15 and C13.
Yeah.
Bullet 2. We found carnivores have way more of these enterocytes 1.
Right.
And bullet 3. Thanks to the lab controls and the glowing FIH imaging, we know this is a permanent evolutionary blueprint, not a temporary diet fad.
Perfectly summarized.
So here's a question for you listening. Think about your own diet and your own ancestral history. If your cellular makeup shifted permanently based on what your ancestors ate over generations, what specific kind of specialized cells do you think would dominate your digestive tract today?
It's a fascinating thought experiment, and to really understand how that permanent shift happens in the fish, we have to go deeper than the cell membrane.
We have to look at the raw code.
Yes, because the DNA instructions driving these enterocytes 1 are behaving very strangely compared to normal evolutionary rules.
Right, so we are transitioning from the number of cells to the actual molecular mechanics inside the nucleus. And the genes running these fat processing cells are absolute rule breakers.
They really are.
When I was reading through these notes and trying to synthesize why these specific genes mutate so incredibly fast, I realized they all share 3 main traits. I actually started calling them the YSF cells in my head.
YSF.
Yeah, young, specific, and free.
You know, that framework actually maps perfectly to the genetic data in the paper.
Oh, awesome.
But to appreciate why being young, specific, and free matters, we first need to define the rule they are breaking, which is evolutionary constraint.
Okay, explain that constraint.
Usually, genes that control fundamental day-to-day cellular functions are under strict stabilizing selection.
Meaning, if it works, don't touch it. If a gene is responsible for, say, turning oxygen into energy, a random mutation in that code is almost certainly going to be fatal. So that genetic sequence stays frozen over millions of years.
Right, stabilizing selection is biology's quality control. But the single-cell data revealed that the genes expressed in these enterocytes 1 have incredibly high rates of sequence divergence across the different Cichlid species.
They're mutating rapidly.
Very rapidly, they are not frozen at all.
So let's apply that YSF framework to see how they get away with this rapid mutation. The Y is young.
Yes. The researchers measured this using a transcriptome age index, or TAI.
How does that work?
By using an approach called phylostratigraphy, scientists can look at a DNA sequence and trace it back through the evolutionary family tree to see when it first appeared.
Like carbon dating for code.
Sort of, yeah.
Yeah.
And they found that enterocytes 1 rely heavily on evolutionarily younger genes.
Okay, so because these genes are relatively new additions, they haven't been deeply hardwired into those ancient life or death biological pathways we mentioned. It's like writing a new piece of software code for an app. You have a bit more leeway to experiment with it and break things without crashing the entire computer's operating system.
That's a perfect analogy. That's the why.
Now the S is specific.
The specificity is measured by a mathematical metric called a tau score. The genes driving the rapid evolution in enterocytes 1 have very high tau scores, which indicates extreme tissue specificity.
Meaning they aren't multitasking.
Right. These genes are not being turned on in the fish's brain or its heart or its gills. They exist and function exclusively in the anterior gut.
And that extreme specificity sets up the F, free.
Free from pleiotropic constraints, yes.
Okay, we need to detail what pleiotropy is.
Pleiotropy is a critical concept in genetics where one single gene controls multiple, seemingly unrelated traits across the body.
Okay.
So if a gene operates in both the gut to digest food and the brain to regulate hormones, mutating that gene to make digestion 5% more efficient might accidentally cause catastrophic neurological damage.
Oh, man. So that multitasking creates a massive evolutionary roadblock.
Exactly, but because the enterocyte 1 genes are so specific, they have very few protein interactions, or PPIs, outside of their immediate local network.
They're solo operators. They really only do one job.
And we know this freedom leads to actual functional changes because the researchers analyzed the DNDS ratio of these genes.
Okay, we need to thoroughly explain the DNPS ratio because, as I understand it, this is the ultimate proof of active evolution. It's a comparison of two different types of mutations, right? Synonymous versus non-synonymous.
Right. Let's break down the mechanics of a mutation. DNA is read in 3-letter chunks called codons, and each codon codes for a specific amino acid.
Right.
But the code has redundancies. Sometimes, if a single letter in the DNA randomly mutates, the new 3-letter chunk still codes for the exact same amino acid as before.
So the end product doesn't change.
Exactly. The resulting protein doesn't change at all. That is a synonymous or silent mutation.
It's a typo that doesn't actually change the meaning of the word. And because it doesn't change the protein, evolution basically doesn't care.
Right.
It doesn't help the fish. It doesn't hurt the fish. It just acts as a quiet background ticking clock of random chance over time.
Exactly, the mechanism. But a non-synonymous mutation changes the letter in a way that does alter the amino acid.
Okay, so that physically changes the shape or the function of the resulting protein.
Yes, and the DNDS ratio simply compares the rate of those functional changes to the background ticking clock of silent changes.
So what's normal?
In most genes, the ratio is far below 1 because functional changes usually break the protein and get weeded out by natural selection.
But in these enterocyte 1 genes,
The ratio is highly elevated. The rate of non-synonymous mutations is outpacing the background clock.
wow.
Yeah, that mathematically proves that natural selection is actively favoring functional physical changes in these specific proteins.
Okay, let's bring it all together for another recap.
Sounds good.
Bullet 1. We move from the cell layer to the DNA layer.
Yep.
Bullet 2. These cells use the YSF rule. They can evolve rapidly because they utilize young, highly specific genes that are free from the constraints of multitasking in other parts of the body.
Perfect.
Bullet 3. The elevated DNDS ratio proves this evolution is active and functional. So, listener, here's your question for this section. Do you have a highly specific single-task tool in your kitchen or workspace? Like a dedicated apple core.
Or a garlic press.
Right, garlic press.
Yeah.
Notice how much easier it is to bend, modify, or completely replace that one tool without disrupting your entire life, compared to a multi-tool that you rely on to open cans, tighten screws, and cut wires.
That is exactly what evolution is doing here. If a gene only does one job in one cell, evolution can treat it like a laboratory experiment.
Right. But that freedom to experiment raises an essential question about the timeline. Well, we know the cells change. We know the genes have the mathematical freedom to mutate. But how fast does this actually happen? Is this just a slow, steady, continuous drift over 10 million years? What does the actual speed of this adaptation look like on a graph?
It's not slow and steady at all. To model the speed of trait evolution, the researchers applied complex mathematical models to the genetic data. Now, usually, normal random genetic drift is modeled as Brownian motion.
Which is basically a random walk, right?
Yeah.
Like a drunkard wandering around a field. Every step is random, and over millions of years, they might drift far from where they started. But there's no specific direction or pull.
Exactly. But the gene expression divergence in these gut cells did not fit the Brownian motion model at all. The model that fit best was the Ornstein-Uhlenbeck, or OU, process.
Okay, let's unpack the mechanics of the OU process. How is it fundamentally different from that random walk?
An Ornstein-Uhlenbeck process models a system that is experiencing a random walk, but it's constantly being pulled toward a central optimal state.
Okay, give me an analogy for that.
The classic analogy is a rubber band attached to a peg. If the environment is stable, the peg doesn't move. The rubber band allows a little bit of drift, but it keeps the organism's traits relatively stable, pulling them back if they wander too far.
But what happens when the environment violently changes?
The peg moves.
Oh, wow.
Yeah. If a fish enters a radically new environment, say a tectonic shift alters the lake, and the fish suddenly has to switch from a plant-based diet to eating other fish, the optimal state for survival abruptly shifts.
So the rubber band stretches.
It stretches to its absolute limit, and then it snaps the biology toward the new optimum very rapidly. Once it reaches that new optimum, the rubber band stabilizes again at the new peg.
So it's not a slow, steady ticking clock. It's an explosive burst of intense evolutionary speed followed by a long plateau of stability.
That's it, exactly. The data shows massive, explosive bursts of accelerated evolution in these specific genetic pathways. The paper actually highlighted a specific group of cichlids, the Trophini tribe, where this is incredibly obvious.
What happened with the Trophini tribe?
About 1.5 to 4 million years ago, the water levels and the physical environment of Lake Tanganyikan fluctuated wildly. Major new ecological niches just suddenly opened up.
Chaos in the lake.
Total chaos. In response, the Trophic tribe experienced a major pulse of dietary diversification. As they rapidly switched back and forth between grazing on algae on rocks to hunting other fish, the OU models show massive bursts of accelerated evolution in their enterocyte 1 genes.
They were literally leaping across dietary boundaries, and their YSF cells were mutating and adapting in real-time bursts to keep them alive.
Yes. It shows that when the ecological pressure is high enough and the genetic constraints are low enough, adaptation is explosive. It's perfectly timed with the ecological shift.
It perfectly aligns the internal cellular machinery with the external environmental chaos.
Precisely.
Okay, final recap before we wrap up.
Let's hear it.
Bullet 1.
Yeah.
We move from slow, steady drift to the OU model, the rubber band. Bullet 2. The Trophic tribe proves that this happens in rapid bursts driven by ecological chaos. So, to the listener, what environmental pressures in our world right now might be forcing sudden, rapid biological shifts in animal populations today?
It really makes you think about how dynamic life actually is.
It's fascinating. And, it really brings us to the core, so what, of this entire data set. Why should you, listening to this, care about the mathematical mutation rates of African fish intestines?
Right.
Because this deep dive proves that ecological adaptation doesn't just happen on the outside. It is multi-layered. When an animal adapts to a new niche, it doesn't just slowly grow a sharper jaw over millions of years. It literally rewrites its internal cellular workforce and its deep genetic code simultaneously, snapping toward a new optimum to survive.
The Cichlid gut teaches us a hard biological rule. Tissues must adapt at the cellular level to handle radically new nutritional profiles. And if we take that rule and apply it to our own species, well, it leaves us with a rather profound, almost unsettling perspective.
Because our dietary environment has just experienced a massive, violent shift.
Consider the timescale. These fish experienced explosive genetic bursts over thousands of years just to digest algae versus fish scales. Now look at the human diet over just the last 100 years.
Wow. Yeah.
We have introduced a staggering amount of ultra-processed foods, refined, dense sugars, and novel chemical emulsifiers that our ancestors never, ever encountered in nature.
We have completely and radically changed the fuel we are putting onto our own internal assembly line.
We moved the peg in the OU model.
We moved the peg.
So if the Cichlid teaches us that survival requires rapid cellular adaptation to a new diet, what invisible evolutionary pressures are we currently putting on our own enterocytes right now? Are human guts currently undergoing their own burst of accelerated unseen evolution to handle ultra-processed food?
And if they are, what is the biological cost of that forced adaptation? It completely flips how you look at the body. We started this deep dive talking about how much we love clear, visible x-rays of a broken arm. We like evolution when it's a bird's beak we can point at and measure.
It's easy to understand.
But the real story, the deepest mechanism of survival, is happening in the dark. It is happening in the microscopic muddy waters of the gut, where cells are quietly shape-shifting, mutating, and snapping to new optimums just to keep us alive.
The most powerful changes in nature are almost always the ones we cannot see with the naked eye.
I couldn't have said it better. Take a moment to think about your own internal assembly line today. Thanks for joining us on this deep dive. Keep questioning what's happening in the dark.